It's telling me we're live. As always, as I always do, I am going to wait just a second until I can see and hear myself on YouTube so I know that it's live and everything is working. If at any point people cannot see or hear me or my guest today, just let me know. I'm still waiting for YouTube to click over. Now it has clicked over. It looks like it is working. Everything looks good. Okay, let's just mute that so I'm not listening to myself because that's annoying. Okay, let's see. Sorry, everybody. My laptop doesn't like having this many things going at once. Okay, hi, everybody. We are live. I'm very excited to be back. Today, joining me is David Neff, and we are going to be talking about Dr. Nathaniel Jeanson, his work on the mitochondrial most recent common ancestor, also known as mitochondrial Eve, which is like some of the worst branding biologists did. Like, we got to get a PR department in here. But the main thing we're going to talk about today is we're going to talk about how some new work that actually David's done that definitively shows that Jeanson's conclusions are refuted by his own data, by the data he uses to demonstrate his conclusions, actually directly refute them. So that's what we're going to get into today. So what we're going to do, the way this is going to work is we're going to take just a little bit of time. We're going to talk about Jeanson's paper. It's a blog post on AAG, but they call their blog the Answers Research Journal, and they pretend it's a peer-reviewed journal. It's not. It's a blog. So we're going to talk about the blog post from 2017, where he talks about this mitochondrial mutation rate and how it proves that mitochondrial Eve, the mitochondrial most recent common ancestor, existed just 6,000 years ago. So we are going to talk about that in general, and then we're going to talk about the problems with it. David has some stuff that he's been going through. I have some objections to this work, but then we're going to pretend all that stuff doesn't matter. And we're going to show how even if you ignore all the problems with Jeanson's numbers and just take everything he's saying at face value and pretend it's all above board, it's still wrong because of the data he uses. And David, just over the past couple of weeks, figured this out by doing some pretty basic stuff that, like, honestly, Jeanson should probably have done at the front end of all of this. So we're going to go through this, and it'll be fun. I don't think it's going to take that long. David has an after show on his channel at 9:45 or whenever we finish. I'm thinking we're going to be good for 9:45 if you want to go over, talk on David's open mic, which I called it an after show. It's not really an after show. It's an open mic, but presumably you may want to talk about this afterwards. So before we go any further, David, would you like to take a couple minutes to introduce yourself?
Yeah, well, thank you, Dr. Deanne. It is a pleasure to be here. And for those of you who do not know me, I do a lot of debates, open mics and discussions on all things related to religion, science, philosophy, and music. I recently got accepted to the University of Maryland, Palmer County, and I will be majoring in biological science at the UMBC starting in fall of 2021. I am very much excited for this. So my channel is David P. Neff. Dr. Dean, if you don't mind, can you make me a moderator so I can post a link to the after show?
Yes, sir. Let me find you in the chat window here, and I will make you a moderator. Let's see. Where are you? Can you just like post something, just say hi or something? Oh, there you are. I gotcha. Hang on. I'm gonna make you a moderator. Add moderator. You're a moderator now, so you can post a link to your show and or your channel. You're good to go.
Okay. Yep. There is the link to the show. Everyone is welcome. Hope to have some really good discussions over there. So now that my introduction is out of the way, I think we can get started.
Yeah. So let's get into this. Here, before we get into the actual nuts and bolts, I just need to clarify for everybody a policy of mine, which is that whenever I'm going to talk about anybody's work, I'm going to invite them on. So you have the opportunity, if I'm criticizing your work, to like defend yourself. I'm not gonna say like so-and-so is wrong, it was terrible work, blah, 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 blah. If they're, without giving them an opportunity to explain like why they're actually doing good work. So as you can see in this message, this is actually a Facebook message because real scientists have publicly accessible emails, and you can just like email them with any questions you have, as we will see shortly. That's something you can do. That's not the case for Nathaniel Jeanson. So you have to get to him via Facebook message. For what it's worth, you could just email AIG, which I did, but they didn't get back to me. But it's just kind of the general like inquiry email for them. It's not a thing for Nathaniel Jeanson. So I just said, you can see it's very polite. I just said, I'll be discussing your work on the mitochondrial most recent common ancestor, blah, 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 blah. I always invite people on. Here's my email, and this is my channel email. So there you go. Anybody can email me. That. Oh, actually, that's my other. Oh, that's the wrong email. Whatever. Doesn't matter. It's fine. People have that email too. It's cool. So then, just to be sure, cover all my bases, there we go. I sent him the link to this show right now. So, and as you can see, he saw it. He's seen these messages. He's going on Facebook and he's seeing them. Whatever. So, you know what? He had the opportunity to be here. He didn't want to be here. I invited him. I would love to talk to Dr. Jeanson. I think that'd be really cool. Either he couldn't or didn't want to be here, but he didn't respond to say like, sorry, I can't be there. But, oh well, what you going to do? So, just very briefly, what Jeanson claims is, and this is the post, a young Earth creation, human mitochondrial DNA clock, whole mitochondrial genome mutation rate confirms D-loop results. He calculates a most recent common ancestor, that is the convergence point in the past at about 6,000 years ago, and he does this using a thing called the pedigree mutation rate. Now, a pedigree mutation rate is when you look at parents and offspring and you look at the differences between them. That's a pedigree mutation rate. Like a pedigree is you got grandma and grandpa and you got the parents and then you got the grandkids, right? That's, you look at the different, you could track like genetic disorders through a pedigree. So he uses this generation over generation mutation rate. How many changes, how many differences are there per generation? And then, extrapolate that backwards. And you get your time to most recent common ancestor based on how many differences there are. Now, the data he uses, I say Jeanson's data because he didn't collect the data. He's using data from other studies, and he referenced 3 studies, but he actually only really referenced 1 because, as you can see right here, the mutations he looked at, 2 of the studies had 0. One of them, Ding 2015, had 63, and those are the mutations that we're dealing with right here. Okay, so these 63 mutations in these pedigrees, these parent-offspring surveys, were the basis for Jeanson's conclusion. So those are the data that he's using. He's looking at parents and offspring and the number of differences. And the one study he's using documented 63 specific mutations. Right? Now, the way you calculate, don't worry, Neff, it's right after this, then we're going to you with all the stuff that you have lined up, because just you wait, everybody. So the way you calculate the time to most recent common ancestor, and that's what this is, TMRCA, time to most recent common ancestor. What you do is you determine the long-term mutation rate based on known divergence times. So like for viruses, which is what I'm familiar with, really familiar with, is like we know when a specific virus jumped into a new host, right? Because we know when this disease emerged. Okay, so now we can use that as the known divergence time, and we could look over tens of thousands of generations, which may only be a few decades for viruses, how many substitutions accumulate in that lineage. And then we could look at the most divergent samples for that group of viruses overall. Take the 2 most divergent ones and say, okay, given that rate that we've observed, let's see how long it would take to accumulate all of those differences. Now, it's more complicated than that because you have to deal with things like population size and stuff. It gets more complicated, but that's the idea. You document the long-term rate based on a known divergence event, and then you apply that rate to the largest number of differences you can find within your sample group to work backwards to see where the time to most recent common ancestor falls for that group. That's what we're basically doing here. Now, Jeanson doesn't do this. There's so many things wrong with what Jeanson actually does. As David said earlier, every time you look at this paper, it just gets worse and worse. And it's not even a long paper. So at this point, I'm going to turn it over to David Neff. And David, you're probably going to want to share your screen. So I'm going to stop mine and take whatever time you want, go through the various problems that you have accumulated with this paper. Then I'll talk about my favorite problems with this paper. And then we'll talk about the new stuff that even if you put all that stuff aside, it still invalidates this paper. If you assume nothing that anyone has ever said about this paper is wrong, if you Assume all that stuff. My criticisms, everything's wrong. We're going to talk about why it's still wrong. So if you want to share your screen.
Yeah. I'll share my screen in a minute. But I want to begin with a very simple observation. And that is that multiple independent teams of scientists have worked on the problem of calculating the time to the most recent common ancestor. And Dr. Jeanson is the only person to get the result that he did. So the fact is, nobody has been able to replicate his results. And as anyone who is familiar with science should know, if your results cannot be replicated, then that is pretty much an alarm bell saying, hey, my results are probably wrong. Just to bring this home a little bit, Dr. Dan, what is the largest class that you have? What is your largest class?
The largest class that I teach?
Yeah.
It is almost 500 people. Largest class I teach gets about 480 people every fall.
Okay, so let's just round that number up to 500. If you were to give a test, there are 500 ways for the class to get a question wrong, but only one way to get the question right. If 499 people came up with one answer and one person came up with the other answer, there's a very good chance that the one person is incorrect and the majority of the class is. And that's one of the things that I want to demonstrate is that all of the other, if we were to plot out do like a data plot of all the studies that have done mtDNA and figure out, okay, we're going to do a data plot of where they fall. Dr. Jeanson would be the obvious outlier in that study.
Yep. So it would be Jeanson over here, and then everybody else would be over here somewhere. There'd be a big distribution over here, and then Jeanson all the way down here, smushed in by 0. Yep.
Yep, and that is basically how we would look at it when we were to do a data plot, if we were to do what's called a meta-analysis of the studies that have done this work. And that is where, when you actually, and the thing I love about meta-analysis like this is that is when you really start to find, okay, maybe there's an error in this paper over here, when you compare that to all the other papers that have been published on that topic. The second problem that I see is that Jeanson's sample size is way too small. In the Dang paper, they used only 333 pedigrees. The others were 26 and the other one was 39, so that's a total of 398 pedigrees. Of that, total 6,594,462 base pairs were examined, and only 63 of the homoplastic mutants were detected. So I'm going to share my screen here. If I can pull that up. So this is his data here. And if you look at the ethnicity, we see Danish, Finnish, European, and Sardinian. So the whole entire data is pretty much centered around the European ethnicities. This is simply not enough data. And the kicker is Dr. Jeanson knows this. How do I know he knows this? Because he later says the following, if I can find it.
There, I think it's that paragraph right there. Conversely.
Yeah, here we go. Conversely, since certain African groups are more genetically diverse than non-African groups, it remains to be seen whether the results in this study will hold true in the African groups as well. Now let's stop here for a moment and let's use our brains. Why didn't Dr. Jeanson include African DNA in this study? If he is basically openly admitting that we don't know what this results are going to be holding true if we include this analysis, then why didn't he include African DNA? I asked Dr. Jeanson this, and he did not answer me.
Can I just jump in for 1 second with a small observation here?
Oh yeah, go right ahead.
If I'm reviewing a paper, and I've reviewed papers as part of the peer review process, if I'm reviewing a paper and the author says, now we have a conundrum here, we don't know if it's A or B, and there's a simple way to test that, what I'm going to write back is like, you should not leave that hanging, and you should not just assume it's one or the other. You should test that because we have the data to do that. So, like, you should look into that and either find a reference that has done that or collect the data and do the calculations yourself. That's the obvious thing to do right there. Hey, I've got a question. I don't know which one it is. Like, okay, doctor scientist from Harvard, like, do the science, figure it out. That's just just do it.
Yeah, exactly. And let's go on, just so I won't be accused from standing for truth with quote mining. I'm going to read what he says after that. Two major hypotheses exist for the increased mtDNA diversity in Africans. Either they originated first, or they mutated their mtDNA slightly faster than non-Africans. So let's digest this information real quick. There is something called the founder effect. And if you were to take a look at the human population and figure out, okay, Africans have the most genetic diversity, we can predict based on that alone that humans originated in Africa. Because when you have a parent population and then you have a small population that goes off somewhere else, that small population is losing the genetic diversity from the parent group, and so they are going to be less diverse genetically. If I can do an analogy real quick, imagine that we establish a colony on the moon, and then from there, the people on the moon, a small portion of them, establish another colony on Mars. If we were to do a genetic analysis, by understanding the founder effect, Earth would have the most genetic diversity, followed by the moon, and followed by Mars. This really is not that difficult. So the second thing is that they mutated their mtDNA slightly faster than non-Africans. The problem here is that there's simply no evidence that they mutated their DNA faster. Moreover, there's no mechanism by which it can speed up or slow down in this group, but not over here in another group. And let's continue. The evolutionary out-of-African model naturally holds to the first explanation, but the results of this study already undermine this hypothesis. Now, this first statement will be pretty much repeated later on in this video. The most diverse humans in the mtDNA database are 123 base pairs apart. Yet after just 50,000 years of mutation, non-African humans would have been at least 170 base pairs different. Had we used the evolutionary date for the origins of the African people groups, e.g. 180,000 years ago, the predicted amount of mutational differences would have been even higher than 170 base pairs. Hence, the evolutionary model needs slower mutation rates overall, not just in non-African people groups. So, of course, the problem there is that his methodology is simply wrong, which we will talk about in a little bit. But there's an even bigger problem than all of that. Even if we concede the point that maybe Africans did in fact mutate a little bit faster, that still does not explain the fact that when we do a phylogenetic analysis of the genetic diversity, non-African genetics diversity is represented as a subset of African diversity. That is proof that we originated in Africa because we are a subset, a nested hierarchy of the African genetic groups. That's all there is to it. It really is not that hard.
Yeah. There's literally no other way to get that pattern unless the African set of diversity on that tree is the root of the tree. There's no other way to get the pattern of nested diversity that we observe in actual human populations. It's simply not possible.
Yeah,
it's
exactly.
That simple. Like it really is that simple.
Yeah. So here's another paragraph that I want to draw everyone's attention to. Scripturally, all ethnic groups likely find their ultimate origins at the Tower of Babel. Genetically, the structure of the human mtDNA tree reflects this event. No, it doesn't, implying that non-African and African groups originated at the same time in history. Based on the successful based on the successful results of the study, I predict that the most diverse African groups will be found to mutate their mtDNA at a rate twofold faster than reported here. Well, there's his prediction, but where is his test? Where does
Yeah,
he actually
you need to
test
test
this
that.
hypothesis? He simply doesn't.
You got to test that.
Yeah, exactly.
Yeah.
So, and then he says, conversely, the results of this study demonstrate that the current rate of mtDNA mutations in non-Africans is predictable from the young Earth timescale and from the mtDNA diversity among extant humans. Again, he says that this underscores the previous contention that DNA sequences from fossils are too degraded to be useful for comparative DNA analysis today. So, again, there's his prediction, but where does he actually test that prediction? And then this is the fun.
I do want to point out for everybody, this was 2017. There have not been subsequent follow-ups where he goes through and actually collects those data and tests these predictions. They're just out there, and they're not. Oh, sorry, 2015. Even worse. It's been 5 years. It's been 5 years.
Yeah, I think the paper he does as a follow-up with the unrooted tree. Does he do anything by testing these predictions? Since you've read that paper and I haven't.
No.
Yeah, that is deeply problematic. But I just wanted to draw everyone's attention to the length of the paper. This is only 4 pages in length. And if you are doing an mtDNA analysis of calculating the time to most recent common ancestor, your paper is going to be much longer than 4 pages if you are doing the technique properly. And I counted the number of references that he had down at the bottom. It only came to about 17 papers. And others, 17, you see Creation Research Society, you see that Dr. Nathaniel Jeanson cited himself twice, Jeanson 2015 and Jeanson 2013. More creationists,
there are other names. Many of you will recognize them there. There's 2 from Tompkins. There's some Snelling in there. It's all familiar names.
Yeah. I actually don't see Snelling in here.
He's a second author on one of them.
Okay.
Yeah, he's on that last one. Snelling is the second author.
Okay, there we go. The RATE project, which
there you go. Yeah, all right.
These are just basic issues with it.
Yeah.
And we're going to have a lot of fun with this paragraph here. The brief results presented here strengthen the...
Are you looking for the one above it?
There we go. The only remaining caveat to this present result is whether or not the mutation rate reported in Ding et al. Represents a germline rate rather than a somatic mutation rate. To confirm a germline transmission in the future, the DNA sequences from at least 3 successive generations must be sequenced to demonstrate that variants were not artifacts of mutations accumulations in non-gonadal cells. So, he's basically saying, I don't know, it's one way or the other, but I'm going with the other way. Now let's use our brains for a moment and think for a moment. If you were Dr. Nathaniel Jeanson and you were using Ding's study and you weren't sure if it was somatic or germline, what would you do? What if there was a very easy way to get a hold of the author of the original paper and ask him? I know, maybe email him. And spoiler alert, that's exactly what I did. I emailed the lead author of the Ding study, Lynn Ding. I actually emailed them, and they got back to me. And we are going to have a lot of fun with that response later on. So with that, I will turn it back over to you.
Thank you. Yeah, so we're gonna wind up right with that email. So I want to first highlight the other major problems with this paper. So I'm going to go back to my screen here because it gets better. But wait, there's more. What Jeanson basically sets up in that paragraph that David just read is that you have the somatic rate. You have, all the mutations in all the cells in the body, the muscles and the neurons and the stomach and everything else. And then you have the germline rate, the mutations that are in the cells that produce gametes that could potentially get passed on. Now, that is just wrong in terms of calculating a long-term mutation rate because you are doing a heck of a lot more filtering than just somatic to germline. So I used these slides when I talked to Sal Cordova a few weeks ago on this same topic. And I'll just briefly go through them here. But if you consider this oval, this is all mutations, every tissue in your body. Now, the germline represents a subset of that. This is not to scale. The germline does not represent most of the mutations. The germline represents a teensy tiny fraction of the mutations in the body, right? Just think about just in terms of like numbers of cells. How many cells are in the ovaries and the testes compared to the rest of the body? It's just not that many, right? So the germline mutation rate is going to be a very small subset of the total mutations that occur in the body. Now, Jeanson basically cuts it off there and says, but if it's in the germline, you're good. That can persist. No, because you still have to get into the gametes. And this is a problem for both males and females. Not every cell that could make sperm is actually going to ultimately make sperm. Most cells in females in the ovaries don't ultimately become secondary oocytes. Most of them don't. So like, just think about how the ovarian cycle works. You have a bunch of follicles mature every month, and then only one of them is actually going to go through ovulation, right? Sometimes it's two, and that's why you get fraternal twins. But for the most part, it's one. And then the other, like 3, 5, 10, 50, however many there are, those just go away. They're dead, right? So all the mutations and the ones that don't mature, those mutations are gone. So you have a germline to gamete filtering step where you lose a bunch of mutations. Now, of those gametes, the vast majority of those don't go through fertilization, don't become a zygote, right? So again, you have another layer of filtering. And then the zygote, of course, has to successfully grow into an adult for that mutation to persist in the population. And then the adult also has to go through, in the next generation, you got to go through all these steps again, right? You have to, all the mutations in the adult, they have to be in the germline, make it to the gametes, yada, 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 over many generations in order to actually persist in a population. What we care about when it comes to long-term mutation rates, time to most recent common ancestor calculations, what we need to do is use the substitution rate. So here's what Jeanson claims. He says, I use the germline rate, it's cool. He ignores all the levels of filtration kind of below the germline rate. He's just pretending everything that makes it to the germline, all those mutations persist. Yeah, David, go ahead.
Yeah, by the way, I did a Command F on my Mac just to make sure I didn't miss anything. The word substitution does not appear anywhere in his paper.
Doesn't appear at all.
Just wanted to throw that out there.
Yeah. So Jeanson claims he uses a germline rate. What he says is, well, it could be somatic, it could be germline, we're going to go with germline. He says, it's kind of wishy-washy. What he actually counts based on his techniques, and as we'll see, we have an update on this front, is all mutations, somatic and germline, right? What he actually counts is everything. Now, what matters for time to most recent common ancestor are the fixed substitutions. What mutations that occur that ultimately become fixed in the population. What we care about is the rate at which new mutations fix in the population. Now, the key thing here is that because you have all these filtration steps, plus you have natural selection, genetic drift, bottlenecks, right? You have all these steps that weed out genetic diversity. So given a mutation, the likelihood that it persists within a population decreases over time. So the mutation accumulation rate, or being more technical, the substitution rate, the rate at which mutations fix, is way, way, way lower than the spontaneous mutation rate, the rate at which mutations occur. And this is a figure from Evograd's excellent multi-part review / debunking of Jeanson's book, Replacing Darwin. And this shows basically this schematic relationship. So you have the time on the x-axis and the rate on the y-axis. So your spontaneous mutation rate is very high. But very quickly, you start to weed out those mutations due to all the filters that I just described. And as you go through all those filters, you converge at the evolutionary substitution rate, or you sometimes hear the phrase the long-term substitution rate. That is what we care about when it comes to calculating the time to most recent common ancestor. You cannot take a pedigree rate or a germline rate and extrapolate backwards. And I should note that Jeanson conflates those two things, and they're not the same thing, but whatever. You can't take a germline rate or a pedigree rate, which is going to be up here, and extrapolate that backwards over time to get your time to most recent common ancestor. You simply cannot do it. It does, like, that's not how these processes work. It's just wrong. It's not more complicated than that. Now let's assume that none of that stuff matters. Let's assume that everything Jeanson did, let's assume he measured the mutations right, he did the math right. Let's just assume that nothing David just talked about and nothing I just talked about matters. Would Jeanson be correct if we took everything at face value? Nope. And the reason is what we'll get into now. First is that paragraph that David showed you where he says, we don't know if it's a germline or somatic rate. We think it's germline, so we're going to go with germline, right? That's the first place. This is the disclaimer paragraph where Jeanson says, if you understand the language, what Jeanson is saying here is everything I just said, I don't know, maybe, but he says it in a sciencey way. So if you're not, trained in the field, you probably wouldn't catch that. Who knows? So now David actually got a hold of Nathaniel Jeanson, and David, I have it popped up right here, but you want to just tell everybody what you asked him?
Yeah, so I actually had asked him a couple of questions about this, about Neanderthals, and about African genetics, and about a control. We'll get into that in a little bit, but I wanted to kind of hammer in on this paragraph here. And I asked him, just to clarify even further, if Ding used the somatic mutation and not the germline, would your results be invalid? And this is what he said. Potentially, yes, it might be invalid. If Ding et al. 2015 used a somatic rate, then the germline weight would be unknown. However, other independent lines of evidence seem to confirm that it is indeed a germline rate. Now, I want you to notice how careful Jeanson was in his wording. He says independent lines of evidence seem to confirm a germline. Seems. And what is the evidence? He provided none of it. And he basically admitted that, basically he's saying, yes, it would be completely invalid. So if his paragraph in that paper wasn't clear enough, here it is even clearer.
There you go. And the key line here is then the germline rate would be unknown.
Yes.
That is the absolute key, because that is saying straight up, if they caught somatic mutations, then my math is not what I said it is, right?
Exactly.
So that's the key line. So as David alluded to earlier, he went the extra step, right, that you would think like Nathaniel Jeanson might have done in this situation, and asked, Ding, what's up?
So basically the email I asked Jun Ding, I emailed him and one of the other authors. The other author was retired, but he said that Jun Ding would get back to me. I basically asked him, hey, young recreationist, Dr. Jeanson is using your data in this blog post. Number one, do you have any response to Jeanson's work? And number two, did you use somatic or germline in that study? So here is what Ding said. Ding said,
I should probably have that pop up, shouldn't I?
There we go. So he said, thank you for your email and for the questions regarding our 2015 PLOS genetic paper. We have 2 points of response. Number one, in our original PLOS genetics paper, we never tried to estimate mutation rates. It's in Dr. Jeanson's post that he uses our trio data to estimate mutation rate. This work was not done to determine rates of germline mutation, only prevalence and the variance in the samples. So he then says, in this blog post, he used 63. What was that?
Yeah, I just pointed out blog post. It's a blog post.
Yeah, blog post. He used the math, the 63, 35 plus 28 mutations from our data set to estimate mutation rates. We believe that these 63 homoplastic mutants could represent a combination of germline and somatic mutations. So that is the kicker. That is the slam dunk right there. The final nail in Jeanson's coffin is Jun Ding basically saying, hey, we're not even trying to determine germline mutation, and this is probably a combination of both. So, translation, from Jeanson's own admission, we cannot know the germline rates.
Which means everything Jeanson said is wrong. And I want to point out the very specific thing that's the key here. We believe these 63 homoplasmic mutants could represent a combination of germline and somatic. Could represent. The reason they're saying could represent is it's a very particular word choice here. The reason he worded it that way is because they didn't do anything to distinguish in their data set. So they're not saying whether any one of those mutations is germline or somatic. They're saying it could be either. We did not do anything to tell them apart.
Yeah, and right, and
basically what said came to pass.
Yeah, and basically it could represent a combination of germline and somatic because the whole work was not done to determine rates of germline mutation.
Yeah. Yep. You can't use these data to determine a mutation rate.
Exactly.
These type of data are inappropriate for what Jeanson uses them for. It's that simple. You cannot use them in the way Jeanson used them because the authors that collected these data and wrote about these data and got them published, they did not do the things that would be required to be able to tell enough about them to do what Jeanson does with them. And here it is in black and white, Ding saying, like, straight up, we didn't tell them apart. We don't know. They could be one, they could be the other. We can't use them for mutation rates. Like, that's the ball game. So I just want to summarize this really fast, just to review what we've talked about. Jeanson is wrong about everything here. So he ignores selection, right? He ignores drift. He ignores all the other mutation filters I talked about, right? He ignores bottlenecks, right? And David describes some of these. A founder event is kind of a category of a bottleneck. It's a similar kind of dynamic, right? He ignores that other parts of the genome have a larger effective population size, which means even if everything about the mitochondrial DNA is right, let's assume that everything that David and I have said so far is wrong. Let's assume that. Jeanson is still wrong because the other parts of the genome have a larger effective population size, which means the convergence date is further in the past. So if you put the mitochondrial convergence date at 6,000 years, the nuclear DNA is necessarily going to go back further than that, and that violates the Eck timeline. You can't squeeze all that stuff into 6,000 years. But let's put all that, I couldn't even fit all this on one slide. Let's put all of that stuff aside. And yeah, I see in the chat, rate heterogeneity. Yeah, let's put all of that stuff aside. Even on the one small thing, the you had one job version of this, right? Using the data from Ding 2015 to determine the mitochondrial germline mutation rate. Jeanson couldn't even do that correctly for the reason that Jeanson himself identified in the paper where he claimed he could do that. It turns out that the thing that he said, by the way, this might invalidate everything I just said, it turns out that that thing is actually the case, and he couldn't be bothered to check ahead of time.
Exactly. And obviously he didn't bother to do his homework. I emailed Ding, and Ding was very kind to respond, and I really thank him for that. Why couldn't he do that? Why did he have to say, maybe it's this, maybe it's that? I'm just going to go with this because it helps my case.
Yeah. So
if I were to wager a bet, I have a prediction, and my prediction is that he deliberately left out the African genetics and the Neanderthals, and he deliberately used this study or misused the study because he knows that if he included that, then his own analysis would be refuted.
Yeah, we didn't even talk about Neanderthals and how that blasts the rate too, because now the Neanderthal convergence date has to go further back. It's just a big giant mess. So the takeaway from all of this is that Jeanson's own data, the only data that he uses in this groundbreaking, paradigm shifting study, right? His own data refute the conclusion that he drew using those data. That mitochondrial Eve existed 6,000 years ago.
Yeah,
that's the long and short of it. And to be perfectly clear here, the reason we can say this so confidently is because David took the time to do the legwork to actually reach out to the relevant people, right? Jeanson and Ding, and got Jeanson to say, yes, this would be a problem for my work, and then asked Ding, and Ding said, yep, that's the case. Case closed. That's the ball game, ladies and gentlemen.
Yeah, that being said, since we have a little bit more time, I don't want to leave any stone unturned, so to speak. So I want to kind of go back to some of the stuff I said or previously about the lack of the control and some of the issues with Neanderthals. And the thing is, not only when you have Neanderthals, you'd also have Homo floresiens, erectus, nolini, or any other hominin that you want to include as fully Homo sapien. We know that they were far more divergent than we were. They were very much genetically distinct. And there was definitely, we don't have Homo erectus DNA, but their morphology seems to indicate that they were pretty diverse from each other. So that's a lot of diversity that you simply cannot get in a 4,500 year time frame. But I
Yeah.
do want to note that um, even if everything was correct, the 6,000 year time frame is still too old because he needs to be at 4,500 years, not 6,000 years. But let's go on, and I'm going to read some of the emails that I sent to him. So I obviously asked him about the issue on Neanderthals and the control. So let's begin. mtDNA, here's his response to me for the control. For mtDNA for mammal kinds, in theory, yes, it should be possible to do a similar calculation. I discussed the 3 non-human vertebrate species with measured mtDNA mutation rates in my book, Replacing Darwin. It's a longer discussion, otherwise I describe it here briefly. You already are doing a review on that, so I'm going to defer that to you and others. But about germline mutation, he said it would be invalid. And Neanderthal DNA, I discuss it more in this paper and the back and forth that he had with another paper. So apparently someone else on the Answers in Genesis website were also kind of doing a back and forth. I obviously don't have time to read every single thing word for word, but I actually did ask him about genetic diversity and showed him the line from Ding and All saying that this could represent a combination of both. And of course, Ding saw the message according to Facebook, but
Not
he
ding.
did not
Jeanson saw the
respond.
message.
Yeah, Jeanson
Jeanson
100%
saw the
saw the
message according to
message.
Facebook. Yeah.
Yeah, he did. So I'm going to
No
kind of
answer.
go off on a little bit of a bunny trail real quick, just because I wanted to just throw this in here. We can use the DNA analysis to determine when two species kind of split off. And creationists actually do accept this. And what young earth creationists like Jeanson should be doing is using these data to show that the mammalian kinds splitting off around 4,500 years ago, give or take. So I am going to share this real quick. Um,
I got you covered when it pops up.
awesome. Cat family. This is from CMI. Oh boy, you're gonna have a kick out of this. You're gonna love this.
Oh no. Oh no.
You're gonna love this. Sure. So Figure 3A, the radiation of modern cats in the Miocene, and here is the phylogenetic data that they use. So can see that from creationists, they are accepting this. And you can see the cat ancestor right over here on the left-hand side. And you can see that
Oh, they ignore all the extinct cats.
yeah, yeah, exactly. But it gets better. Here is the hilarious part to it. When I find that.
You want me to take it down?
No, not yet. Actually, yeah, you can take it down.
Sure. Yeah, and then if you want to find what you want, I'll put it back up.
Yeah, exactly. Why can I not find this? Here we go. Late Miocene science.
Oh, hey, Neff, can you just check the author on that cat piece real quick? People want to know who it is.
Oh, I am so sorry. This is Barnabas Pendragon and Nico Winkler. I have no idea how to pronounce his name, so I am sorry. I'll put it in the private chat.
There was people were placing bets in the chat, but sorry, everybody.
Yeah, so here is the kicker. They actually got that graph from a actual peer-reviewed paper in Science. I want you to see how they modified it.
Uh-oh. Uh-oh. Let's see this one.
That is where they got the paper. This is from the actual paper that they got it from.
Oh, they cut off the out group, didn't they?
Yeah, not only did they cut off the out group, they also magically erased the time scale that contradicts the young Earth worldview.
That's a
So,
no-no.
yeah, this is blatant academic fraud 101. So they're basically doing some editing. They're getting rid of the out group that they don't like, ignoring the time scale. And so creationists accept that we can use
Yeah.
DNA to do this type of work. And we can use that empty DNA and Y-DNA to figure out, okay, when did lions diverge from tigers and so forth. And
That's such a perfect illustration of the problem, because they'll accept the techniques when it's helpful, but they'll just pretend the dates don't exist.
exactly.
We'll
So,
just pretend that part never happened.
yeah, exactly. And that is the ultimate problem with young Earth creationists. And in fact, if you all go over to my channel and hopefully subscribe to me, I am working on a brand new series entitled How
Subscribe.
Science Refutes Young Earth Creationism. Basically, the problem that young Earth creationists have is that every single field of science, from astronomy to climatology, independently refutes young Earth creationism. And so, I don't think there's any other pseudoscientific theory out there that is refuted by everything. Yeah, and I'm going through each of the fields of science and show how how it refutes young Earth creationism. So I guess to lead out a summary of everything we spoke about, to summarize, lots of problems with Jeanson's work, no control, not enough data, poor sample size. Nobody has been able to reproduce his work, and improper techniques and
Left out all the mechanisms that filter mutations, just pretending those don't exist.
Yep,
Conflating germline and pedigree mutation rates, which are not the same thing.
exactly.
And as we learned, bring it home. This was your headline, so you bring it
yeah,
home.
yeah. So yeah, basically, he is refuted by his own data. So basically, the punchline here is that Dr. Nathaniel Jeanson pretty much refutes himself. That's it. That's the ball game.
Yep.
So everyone, please. I put a link to my after show. Not really an after show, more of an open mic. It is in the sidebar chat. So everyone, please come over to my channel, and we can continue to discuss these topics. And I hope to see you I hope to see every one of you guys on the live stream. So that is something to look forward to. I will see you over there on my channel, and I will be mirroring this video over to my channel as well. So that's a lot to look forward to. Thank you very much, everyone. And thank you, Dr. Dan, for being gracious to host me this evening and for getting this stuff out there. Oh, and by the way, congratulations on hitting 500 subscribers.
Hey, thank you. Yeah. So, David, thank you so much for coming on. And thank you for doing the legwork of actually digging into what these data mean and what you can tell from them and what you can't. That's an important part of how science works, and it's a step that Nathaniel Jeanson left out, which we now know 100% crystal clear, because David took the time to actually do the work that Jeanson should have done, which is fantastic, and I'm thrilled by that. So great work, David. Thank you so much for coming on and telling us all about this little odyssey here, which ends at Jeanson's work actually being somehow worse than we all thought.
Yeah, exactly.
I think it's a good place to end things. So,
I will say that when I started this project, I was not expecting to be so successful. I did not expect to hear back from either Jeanson nor Ding, but I was surprised. I really am surprised that I got back from both Ding and Jeanson. So there you go.
I will say that real scientists are very happy to respond to questions like that.
Yes, exactly.
Like,
And
totally.
if Jeanson didn't know, instead of playing the guessing game, he just go right on and email the original paper and see for yourself. It's not that hard. It really is not that hard.
100%. All right, everybody. So I'm going to wrap this up. You should head over, if you want to continue this discussion, head over to David's channel. He linked the show in the chat. I linked his channel directly below in the description. If you're not subscribed to him, subscribe to him. And yeah, thank you all. I hope you all enjoyed. If you haven't hit that like button for me. And I'll see everybody soon. Have a great night.
All right. Bye-bye, everyone.